Hi, I'm Dr. DNA, and I'm here today to talk to you about one of the most exciting scientific advancements of the last 10 years, the gene editing tool known as CRISPR. But before we get into the details of what CRISPR is and what it can do, first we must backtrack to see how far we've come since the first days of genetic engineering. Genetic modification has been occurring for over 10,000 years, since humans and wolves began to coexist, and before the eventual domestication of the canine species provided enough evolutionary pressure for them to evolve into dogs. Farmers quickly learned to selectively breed their livestock and crops to create offspring with the highest possible yield. Skip forward to the 20th century, when scientists began to insert pieces of DNA from one species into the genome of another. The first genetically modified food to go commercial was the Flavor Saver tomato in 1994, which had been modified to reduce the production of an enzyme that causes fruit to go soft and lose flavor. Nowadays, we have see-through frogs, glow-in-the-dark mice, super muscly pigs, and bigger, more flavorsome salmon. But how did we get here, and what is this DNA that seems to be so important? DNA is short for deoxyribonucleic acid and is the molecule that codes for all life on Earth. It's what makes you you and me me. It remains the same across every cell in your body apart from sex cells, so that the cells can work in unison to achieve the ultimate goal of keeping you alive. DNA is made up of a sugar phosphate backbone and a sequence of nitrogenous bases, which act as the letters that make up the genetic alphabet. It is these bases, adenine, thymine, guanine, and cytosine, that code for the construction of proteins that ultimately determine the phenotype of an individual. When cells in the body divide, it is crucial that the DNA in the daughter cells matches that of the parent cell exactly. Otherwise, a mistake in the genetic code may lead to the production of the wrong protein, which could have drastic consequences in maintaining normal bodily function. Luckily, humans have a very precise replication process that ensures the correct bases are replicated. DNA is then copied onto a strand of RNA in a process known as transcription. The RNA template is then read by a complex of proteins and enzymes to translate the genetic code and attach amino acids to an ever-growing chain. Eventually, the chain is cut and a protein that fulfills a specialized role within the body is synthesized.